Welcome, everyone, to Frontier Night 2022. More than ever, it is really so good to see all of you here, all the faces, all of us real people together, gathered together in person. And for those of you joining us online and around the country, we're so glad to have you with us. We have had a glorious day for weather in St. Louis today. So if you're in some other part of the country, well, we hope that weather moves your direction. I'm Susan L., Vice President and Executive Director of the Foundation for Barnes-Jewish Hospital. Our foundation is so proud to support Siteman Cancer Center and the groundbreaking research that our brilliant physician scientists are leading every single day. We're here tonight to offer you an exclusive look at the people and science behind the spectacular progress being made in cancer research, treatment, as well as prevention. Tonight, you're going to hear about just some of those discoveries and the promising research happening at Siteman right this minute, right here in St. Louis. All of this is fueled and propelled by your generosity and funds raised at our annual Illumination Gala. Each year, the Illumination Gala raises vital support for cancer research, highlighting the importance of the Cancer Frontier Fund. And it is my privilege now to introduce you to one of our lead ambassadors of the 2022 Illumination Gala and Foundation board member, Susie Sachs. Susie, your personal commitment to supporting cancer research, as well as your dedication to continue the fight, means so much to us all. Susie Sachs.
Thank you, Susan, and thank you for joining us tonight in person. For those of you that don't know me, I'm Susie Steinbach Sachs, wife of Jonathan Sachs, here tonight, and mom to Rebecca, Jessica, and Andrew. Together with my dad, Ken Steinbach, I brought a picture of him, are excited to be the first ever father-daughter team to chair an Illumination Gala on June 4th at the Ritz. Plus, you can't forget my mom, Marilyn Steinbach, cheering us on. Siteman's research is a lifelong to everyone facing cancer today and those who face it tomorrow, something my family and I know about firsthand. When I joined the Foundation for BJH board, I did not realize the importance of donations for future research projects. Without your dollars, our Siteman doctors would not be able to do trials, leading to national clinical trials that ultimately progress us towards new life-saving treatments. Without these gifts, our progression to make cancer a forgotten disease would stall. Trust me, this is why giving to the Cancer Frontier Fund is beyond important. Your gifts today change lives forever. Sadly, my dad, picture, Ken Steinbach, is facing cancer right now and cannot be with us tonight. He is cheering me on in the virtual world. Hi, Mom. Hi, Dad. Both of us taking this topic of philanthropy for cancer research very seriously and would love you and all your connections to join in our fight today. Please take a moment to think about all the patients facing cancer. To all the Siteman doctors and nurses that are so committed to cancer research and for being here to share your passions, thank you so much. Let's cheers tonight to, I don't have my drink with me, to a cancer cure in our future. It's now my pleasure to welcome Dr. John Lynch, President of Barnes-Jewish Hospital.
Thank you, Susie, and of course, thank you to your father, who is sort of a Hall of Famer in ambassadorship for this program. So thank you very much.
You.
Yeah, nice to see you. And isn't it nice to be here without our masks? Being able to see the smiling faces for the first time in about 2.5 years or so. When I put my suit on today, and I had to put a suit on, and I had to wear my foundation tie, and I had to put this thing in, whatever it's called, because I knew Tim Eberlein was going to get all dressed up. But it's impossible to match Tim in his dapper outfits. So anyway, but it's great to see you all and to be here tonight. Of course, we're going to hear later tonight from 3 terrific physician scientists from WashU and Siteman Cancer Center. But we're here to celebrate all of you and those that went before you as ambassadors for the Cancer Frontier Fund. Just some facts about it. You all have helped to raise over $35 million over the past decade or so. Those funds have funded over 140 projects, and this is really high-end science. You're going to hear about it tonight. Really amazing, world-class research being done here. What's also amazing is Tim and all the investigators at Siteman Cancer Center and WashU are able to parlay those dollars 5x, 6x, to bring in additional research funds. So another amazing fact about what the Cancer Frontier Fund is able to accomplish for all of us. Why has it been so successful? So I was given some prepared remarks, and I think the foundation folks know that that's futile. So I actually, and Bob knows too. So there were really 3 success factors. It's really simple. First, it took a very simple but bold vision from a leader, Al Siteman and his wife, who's here today with us. Thank you so much. It was simple. It was simple. Build a cancer center that rivals the best in the world so that none of us have to travel for cancer care. They've done it 25 years later. It took a cancer center director, wasn't his day job when he first got here, to actually pull off that amazing vision, that bold vision. Tim Eberlein, thank you for 25 years of leadership and all the colleagues that you helped bring here and fulfill that vision. Finally, the reason why we're here tonight to celebrate all of you and those that went before you. The generosity of our community has been amazing. And it really is the final success factor. So how about another round of applause for Tim Eberlein and his amazing leadership at the Cancer Center for 25 years. I think some of you may know that Tim, after 25 years of his day job, is actually going to hand the keys over to a new colleague. But can you imagine what he's going to be able to accomplish as the director of the Siteman Cancer Center, doing it full time now? So again, congratulations, Tim, and thank you. Tim, would you introduce our guests this evening? Thank you.
So, John, as always, remember, don't?
I know.
Little pouchette. A pouchette, okay? All right, so it's a real pleasure to share the evening with all of you. I want to build on what John just mentioned. None of us would be here if it weren't for all of you. And I think one of the really unique things about Siteman Cancer Center is the relationship that we have with all of you, our community partners, because you entrust in us the fundraising that you do each year, and we carefully invest those funds, and they end up resulting in new immune therapies, in vaccines, in new imaging agents and technologies, different treatments, and even ways to prevent cancer. And ladies and gentlemen, that's what keeps Siteman Cancer Center at the cutting edge. And so that was Al's vision. Why would my friends and business associates feel the need to go elsewhere? Why couldn't they stay in St. Louis? In fact, better, why wouldn't our national business associates come to St. Louis? So tonight, you're going to get to look underneath the hood, as it were, and see what really makes Siteman Cancer Center a special place. Because tonight you're going to hear from 4 really world-class investigators who make Siteman Cancer Center unique. And so, without further ado, I'm going to introduce our first speaker tonight, Dr. Dineo Khabele, who is the chair of OB/GYN, but she's a specialist in ovarian cancer. And I remember when Dean Perlmutter and I were chairing a committee to look for a new chair of OB/GYN, Daneo happened to be the first candidate that we interviewed. And when she walked out of the room, David and I looked at each other, and I said, you know, David, we could save ourselves a lot of time. Why don't you just follow her out the door and offer her the job? So we were really fortunate that Daneo is here. Daneo?
Thank you, Tim. Hopefully everybody can hear me okay. I'm totally blushing. Thank you so much. What he doesn't say is that was the first round of interviews, and there were 2 additional, more rigorous rounds of interviews after that to get to finally come to WashU. I started in June of 2020. I'm the chair of the Department of OB/GYN. I'm also a surgeon scientist, and I specifically talk about being a surgeon scientist because the road to becoming a surgeon scientist with a basic and translational research lab is quite challenging. I have to keep up my skill as a surgeon and also run a lab. But what drives me and motivates me is that I work on ovarian cancer. Ovarian cancer is the deadliest gynecologic malignancy. Patients I see come to me with stage 3 and 4 disease. It's already spread. And, but interestingly, our chemotherapy agents, after performing radical surgery, actually work in about 70% of patients, even with advanced stage disease. But I've got to tell you, when I first finished my fellowship, which was in 2001, we were telling people that they maybe had a year to live if they had a diagnosis of advanced stage ovarian cancer. Fast forward 20 years, we're telling people who have a good prognostic disease under the microscope and molecular features of responsiveness to chemotherapy. Some of those patients are living 10+ years, and dare we say that we may be moving towards a cure for ovarian cancer. So this is, so this is why I do what I do. But I'm also a little bit stubborn. So my research doesn't focus on the types of tumors that respond to chemotherapy. My research focuses on the types of tumors that are resistant to chemotherapy, so we can discover new types of treatment for those patients who have that type of tumor. So what my lab does is uses a combination of looking at human and mouse ovarian cancer cells and growing them in little petri dishes. We also inject them into mice and treat them with new drugs. And then we test some of the molecular features, genetic and protein features in our lab at the bench, and then test that in human tissue samples and see if what we're finding out under the microscope and with our molecular profiling matches what we see in patients. So what's really critical is the infrastructure for this type of research requires patients being involved and being very generous in giving up some of their tissue samples as well as their clinical data so we can figure out what happened to them. But also the infrastructure to grow tissue in tumors in mice, and also to do the molecular profiling and the data analysis of all the different genes and proteins that may be altered by these new drugs. What's so exciting about being at WashU and Siteman is the infrastructure exists here. It exists here because of Frontiers. It exists here because of you. It exists here because we have physician scientists and researchers and PhD scientists who have built up the infrastructure. So it was really easy for me to move my lab here. We are so excited because we finally have multiple gynecologic oncologists, not just me. This is the first job I've ever worked where there are other people like me doing this work. So you have to have a critical mass of people to build on this. And that is in part because of your generosity, that we have had researchers like Dr. Catherine Fu, who has built up an incredible infrastructure here and is doing similar work in trying to understand chemotherapy-resistant ovarian cancer. We now have recruited Dr. Maggie Mullen, who trained here at WashU as a gynecologic oncologist and now is following in our footsteps as a surgeon scientist studying chemotherapy resistance in ovarian cancer. We also have 10 gyn-oncs here, clinicians, all of us see patients, who are enrolling people in clinical trials. And we are leaders in the country, always the top 3 accruer of patients signing up for clinical trials where we're testing new drugs that will hopefully advance and potentially cure ovarian and other gynecological cancers. And so I wanted to give you a very high-level view of what we do. I don't want to get in the weeds of my specific research, but you have to understand that ovarian cancer research and gynecologic cancer research is very severely underfunded on a national level. I'm one of the few surgeon scientists who actually has NCI and NIH funding for my work. And most of the work we do around the country relies on philanthropy and foundations to help support it. So I am forever grateful that you all have been so very generous. I'm really excited to be here at WashU, at BJH, at Siteman, and thank you for listening.
Daneo, thank you. Next is Russell Pochinski. Russell is an associate professor in the Department of Medicine, Medical Oncology, and Russell is doing some really unique immune therapy trials in GU tumors. And so I thought he would talk a little bit about genomics and a little bit about immune therapies. Russ?
Yeah, thank you. Thank you for the introduction. I don't know, should I go over here or walk around? You know, a little bit of a pep rally. Thank you for the introduction, and it's quite an honor to be here. So I am one of the medical oncologists in the Division of Oncology. Clinically, I see prostate, kidney, bladder. I have a lab, and we do tumor immunology. We use mouse models to look at mostly prostate cancer and try and figure out how to treat it better. I put together some slides because I think if I didn't, I would probably just ramble on. I might be a little all over the place. And so just to get into a little bit of prostate cancer background, you know, this is one of the most common cancers out there. So SEER is this organization that kind of, you know, conglomerates all the cancer facts and data and whatnot. So estimated new cases, a lot. Estimated deaths in 2022, a fair amount. So most of these cases are localized, but there is a lot of metastatic disease, and it's still a deadly disease, even though in localized prostate cancer, the cure rates are very high. This seems to be on autoplay, which is fine. And you can see there in 2019, you know, 3+ million men living with prostate cancer. So, you know, the estimates are 1 in 8, 1 in 9, you know, men will get it, and eventually, you know, all men will develop some prostate cancer if they live long enough. And really the backbone since the 1950s has been hormone therapy. So getting rid of testosterone, which is the major fuel for the prostate cancer. So when I talk to patients about the kind of backbone of prostate cancer therapy, it is, oh, there we go. You know, it's like a male menopause. Sorry about this. And it works. It works pretty good, but not fantastic. And so, you know, in our metastatic patients where the cancer is spread, we still don't have cures. okay? So we can palliate patients, they can live several years with metastatic prostate cancer, but we still don't have cures, and that's what we're going for. So that's just kind of the main pathway in these prostate cancer cells of the testosterone driving the cell growth. And so what I'll kind of go through here is some of the work we've done, and I'll pause here to re-emphasize the importance of philanthropy. Each one of these projects has received support from philanthropy, okay, and would not be possible. I am not yet a recipient of NIH or NCI funding. I hope to be soon, but it's very tough. I mean, these are pay lines of 10%, 8%, 6%. They're very hard to get. And so what keeps my lab alive and many other labs alive are support from you guys. We need, especially if you're working on new things that aren't yet kind of standard of care, that support really drives and builds the initial data that you can then use and parlay that into NIH or other funding. So each one of these projects has received support from you guys, so I'd like to thank everyone. So we have been working on what's called a liquid biopsy. So, classically, if you have cancer somewhere, you have to take a little chunk of the biopsy. We have developed a liquid biopsy where you can just take some blood and kind of look at different things. We've also been working on a vaccine immunotherapy where we take part of the tumor and we sequence the genes and make a vaccine from that. So it's a personalized vaccine against your particular tumor. And then my lab in particular is working on something that most people have never heard of. It's a first-in-class immunotherapy, and we'll touch base on that. I'll tell you about it. The liquid biopsy, we named Enhance AR-seq. So AR is androgen receptor. Androgen is testosterone. That's the major driver of prostate cancer. And in close collaboration with Adil Chowdhury and Chris Marr, Adil's in the radiation oncology section, and Chris Marr is in our division of oncology. And really, the confluence of our particular areas of expertise. Adil really is one of the pioneers in liquid biopsy. Chris is one of the premier prostate cancer genomicists in the country. And in this particular project, I contributed the clinical kind of aspects. But basically what we did is we took blood from prostate cancer patients and pulled out the DNA that the tumor sheds, okay? And then we sequenced that DNA and we look at a number of genes on there. And what we found is we could actually predict if patients were going to respond to the treatment or not. Because typically what you do is you say, okay, well, we have this treatment or this treatment. We're going to try this treatment, and we're just going to see how it goes, right? We're going to give you, a couple months, 3 months or whatever. We're going to treat you. And in prostate cancer, we have the PSA, which is the blood test. So if the cancer is shrinking, the PSA goes down. So you try a treatment, and you watch the PSA, and sometimes it takes 2, 3, 4 months before you see a response. And you don't know if someone's going to respond or not. So we developed this liquid biopsy that basically predicts if people are going to have a response or not. So the top kind of blue line there are patients that are responding to the treatment. And the red line that kind of drops off there are people that are not responding to the treatment. Same treatment, same patients. That's what we call progression-free survival, which is the PFS. If you look at overall survival, how long they're living, same type of thing. So we can basically draw blood before they go on a treatment and say you're going to have a response or you're not going to have a response. So if you're not going to have a response, let's try something else. Let's try, chemotherapy or immunotherapy, et cetera. And so, this is really, let's see here, really compared to what we have right now in the field, really one of the best, most promising looking what we call predictive biomarkers. And so just recently, we actually licensed this technology to a company called Tempus. They're up in Chicago, and they're going to develop it and roll it out nationally. So we're actively working on validation studies, and a lot more work is being done. So now on to these personalized, what we call neoantigen vaccines. And this is just a schema of how this is all done. And basically, we take a biopsy of the tumor. We take some normal tissue, which is the blood cells, and we sequence those. So we have tumor sequence and normal sequence. And then, oh, this is on autoplay. You got a little sneak peek there. And basically, you can see here, this was a lot of the pioneering work was done here. You can see here a publication from Elaine in 2017 that kind of goes through this. And this is work with Bob Schreiber and Will Galanders and a whole host of other people at WashU who have developed these vaccines. We have a GMP facility. We can actually make the vaccines at WashU, which is not super common in academic centers. And so we sequence the tumor and normal, and then that circle on the right is actually a representation of a DNA circle, okay? And then we put the sequences, the target sequences, in this DNA circle, and that acts as a vaccine. So you guys, of course, are familiar with the COVID vaccines, which are RNA. So this is DNA, so same type of thing. So instead of spike protein, we have mutated cancer proteins on here, and it's DNA. So then how do we deliver that to patients? Well, we used a system that actually delivers the DNA and then gives a little shock. And the shock actually opens up the cells. There are little holes in the cells, and the DNA circle goes in the cells. And then that kind of expresses these targets and primes and kind of stimulates the immune system. And so here's the first patient on this trial getting their own personalized DNA vaccine. Now, immunotherapy in prostate cancer hasn't worked that well. So what we ended up doing, it's a little busy, but stick with me, is we had to combine it with, it's on autoplay, we had to combine it with a bunch of other stuff, right? So in several tumors, you can use one immunotherapy and get good responses. Unfortunately, in prostate cancer, it's a relatively, what we call cold tumor. And so this is the schema of the clinical trial. And I won't go through all this, but just to say, we use several immunotherapies together to try and stimulate the immune system in these patients. And it took a long time. But again, with a lot of help and input from Bob Schreiber and Will Galanders, we finally completed this trial about a year ago now. We also, jumping up and down, we also received a lot of some funding from the Prostate Cancer Foundation to complete this trial. And we're just presenting the first kind of clinical overview at ASCO that's coming up in June in Chicago. And we'll have a lot of the immune correlates coming in the next year or so. So finally, what my lab primarily works on is a protein called chemrin that I'm certain probably no one in this room has heard of. No one on the study sections has heard of it. So chemrin is actually a relatively newly described protein, and its job is to move immune cells around the body, okay? And immune cells are obviously very powerful. They can get rid of tumors, they can get rid of viruses, et cetera. And so what we found was that when you look at normal tissue and you look at different types of cancer tissue, whether it's a primary tumor or whether it's spread or metastatic to different sites, you can see that Cameron is turned down or turned off in a lot of tumors. And what we've found is that's one of the ways that tumors evade the immune system, okay? Because if you're a tumor, you don't want immune cells coming in and killing off the tumor cells. So we've kind of taken advantage of this in the lab and said, well, if tumors turn it off or down, how do we turn it back up and bring those immune cells into the tumor and shrink the tumor, kill the tumor cells? And so we actually developed a tumor-targeted Cameron immunotherapy. This is a schema of it, and you can see there, one part of it binds the tumor, and the other part of it is the Cameron. The Cameron brings in the immune cells, and over a number of hours, I have made animation on PowerPoint. So, and I will tell you, it's coming up, and there's animation. So what this does is basically, you can give it systemically, and we've done this in mice, and it can target the tumor that has the target on it. If you have a tumor that doesn't have a target, it doesn't accumulate in the target, okay? So it specifically goes to where the target is. It recruits the immune cells into the tumor. It's a platform technology. So, for example, if you have breast cancer and you have HER2 expression, which is a protein on the breast cancer cells, you could use this to target HER2. So it's applicable, potentially applicable to multiple tumor types, which we call platform technology. Here is the animation, and I think it's all right, but I'm sure professionals will be like, that's a little Mickey Mouse. But bear with me. It took hours. So the targeted Cameron is administered systemically, okay? Circulates around the body and only binds tumor cells. Okay, so that is what is happening there. You can see the tumors there with the targets. And then that is bound to the tumor. There are proteins in the tumor called proteases that then will release the Cameron. Okay, that's shown there. The way immune cells get around the body, there has to be a concentration gradient, okay? And that was kind of one of the technical hurdles of why we don't have these treatments already in the clinic. But using this, we can establish that concentration gradient and draw those immune cells into the tumor, okay? And this, we're in the process of using this and looking at current immunotherapies that are FDA approved, like Keytruda and Opdivo, maybe you've heard of these, in combination, because those work pretty well, but one of the big issues with those is if the immune cells are not in the tumor, and they're in the gut, for example, you might get colitis. If they're in the lung, you might have problems breathing, or the skin, you might get a rash. Those activated immune cells are only going to help your cancer if they're in and around the cancer. Okay, so that was it. That was all the animation. No more animation. And thank you again. I can't begin to reiterate how important your support is for all of our efforts. And so thank you guys for being here.
I hope all of you are taking notes vigorously, because we're going to give a little quiz after the next speakers.
So, Tim, was he working on that all day rather than curing cancer?
Actually, he was curing cancer. He was actually curing cancer. All right. The next speakers, we actually have two. Dr. Brian Baumann is an associate professor in radiation oncology And Brian actually is a recipient of an NCI clinical teams research grant, one of only 10 in the country. And Brian is actually in charge of our GU radiation oncology section. And then we have Amir Iravani, and Amir is in the Department of Radiology. And Amir is in nuclear medicine and actually has helped launch actually a new treatment paradigm. It's called theragnostics. And what that is combining nuclear medicine, imaging, with radiation isotopes so that you can get very personalized types of treatments. And this is actually a really hot area. It's actually being led by our Mallinckrodt Institute of Radiology and our oncologic imaging program. And so it'll be interesting to hear the two of them. Thank you.
Good evening, everyone. I think that's working. I'm humbled to be here and privileged to be representative for all the clinicians and scientists at Siteman Cancer Center, which has been working very hard to find ways to fight the cancer. So I'm a nuclear medicine working in the nuclear medicine division within the Department of Radiology. And one of my area of the interest is this novel way of treating and diagnosing the cancer. So this is a quote from Sir Peter MacCallum that nothing but the best is good enough in the treatment of cancer. Because the cancer always finds a way to evade all treatments. And the concept is very actually simple. We have pillars in oncology. Traditionally, we have chemotherapy, surgery, radiotherapy, and immunotherapy, as Russell was touching on, targeted therapy. And this new pillar for oncologic treatment is called theragnostics, which basically cross multiple disciplines in oncology and brings multiple disciplines together from radiation oncology, nuclear medicine, and radiology to form this pillar. I'll take you to a little bit of personal journey about myself, how I did end up to be interested in this field. It was 2015 when I was attending a multidisciplinary team meetings, which is a meeting that all doctors sit there and put their brain together to come up with a solution for patients. And at the end of the meeting, one of the oncologists came to me and said, I've got a patient with the small cell lung cancer. This is a cancer which is very aggressive. And when patient fails to the first line treatment, which is a combination of chemo and immunotherapy, the outcome is really dismal within a few months, if not weeks. And so this patient is progressing rapidly, is in a wheelchair, is in severe pain. Let's look at the imaging, review the image, and see where we can actually radiate to at least give some palliation to this patient. And at the time, I thought, we have a clinical trial open actually for these patients because some subset of this small cell lung cancer express a particular receptor which we can target diagnostically and use it for treatment. And the concepts of what we see, what we can see, we can treat basically. So this patient within 24 hours, we managed to get a imaging which is called Pith, positron emission tomography. And this is how it looked like. All the area in the dark is the area of the tumor, all throughout the bone, causing the pain. So clearly, radiating one of these sites is not going to be helpful for this patient. But the concept is basically what we see in this image, the area in the dark, if we can basically using a molecule which is labeled to an isotope, which enable us to image with our cameras. If we can switch that isotope with a therapeutic one, with a much more stronger radioisotope that actually can treat that cancer and radiate the cancer cells. So that is basically using the same concept of diagnosis going to treatment. And we got this patient, we thought this is patient suitable, actually expressing a lot of those receptors in the body, and within a week the patient got treated with this treatment is administered systemically, and this is how it looked like. So not only we can treat what we see, we can see what we treat. So exactly we know where that dose systemic treatment which administered is gone. And so the area of the dark is all the tumor sites, and you can see there's nothing actually in the normal organs. There's minimal activity within any part of, so none of the normal organs is getting radiation, so its majority is going to the tumor. And just put an example that for one of the lesions we measured the radiation, how much radiation gone, and the gray is a unit in radiation, and our radiation oncologists know that if you're able to give 60 gray to a tumor sites, it's a curative, basically, radiation to patient. And so this is 1 month after treatment, how the imaging looked like. So within a few months, the patient was able to walk and being functional, coming to the clinic. We continue to treat this patient. And one of the triumph for this particular patient was around 8 months, he was able to attend his daughter's marriage. And unfortunately, he succumbed to the disease at around 13 months, but he was able to achieve that. And I can tell you from a perspective of human, reaching those endpoints of being able to, to accompany your daughter in her marriage far exceeds any clinical endpoints in the oncology. And for me personally, being able to accompany the patient in their journey to fight the cancer, it far exceeds any which will be defined as success in academia. So this was an example, but this concept is basically, again, schematically showing here, has been applied to multiple cancers, and actually the concept is not a new concept. It's about 80 years ago in Boston, Dr. Saul Hertz thought the iodine is very specific going to the thyroid. Let's treat patients with radioactive iodine. So the concept started from there, studying thyroid diseases, then developed to other tumors, which is in the small bowel and pancreatic tumors, targeting somatostatin receptors. Dr. Wall, which is in the group as well, he developed the same concept for lymphoma treatments and spearheaded the efforts to use those agents for lymphoma and other hematological malignancies. And most recently, this concept has been applied to the prostate cancer, which is one of the deadliest cancers we have. And this is a picture of a patient that he had, again, a diagnostic pair and then post-therapy pair, targeting the same molecule in the cancer cells. So in 2000, this is a result of another study, phase 2 study, which basically showing 8 patients out of 50 patients we treated. The area in the red is the tumor sites, which has been contoured in red. And this is a follow-up study after patients getting treatment. Of course, not all patients would respond to treatment. We're trying to understand why some patients, despite expressing the receptors, not responding to the treatment, but this is an evolving field. We're just scratching the surface here, but a lot to improve. And it's been increasingly expanded throughout different cancers, and these concepts can be applied to any particular target in oncology. And one important thing is as well, the American Society of Clinical Oncology recognized this treatment in 2019 as one of the 5 advances in the oncology. and it's recognized as a research priority. So I'm here to basically, by collaboration with the different disciplines and radiation oncology and medical oncology, to develop these treatments for bringing these treatments from preclinical studies to the clinical studies. So I'm handing over to Dr. Baumann, my dear colleague from radiation oncology.
Thank you. Great. My name is Brian Baumann. It's a real pleasure to be here tonight to talk with all of you. And I just want to share with you kind of my similar enthusiasm for this concept of theranostics in the treatment of cancer. And I really think that this is going to truly revolutionize how we treat many cancers, prostate cancer certainly among them. That's one of my areas of expertise, but also others like neuroendocrine cancers. And I think that with better imaging, better targeting, we can achieve better outcomes. So I want to first just tell you about an important study that was published recently. And in this study, it was in prostate cancer patients who were treated initially with surgery to remove their prostate. And unfortunately, their cancer then came back. This doesn't happen at WashU. This was done at another institution. Cancers don't come back at WashU. Right, Dr. Eberlein? Right. And in this particular study, patients were coin flipped to either get the standard imaging prior to their radiation to try to salvage them back to a cure, or this new PET scan where they were injected with a weekly radioactive tracer that was tied to a molecule that's very specific to prostate cancer. And then the patients in the conventional group just got our usual radiation. The patients who got the imaging, the special imaging study, their radiation treatments was adjusted based on the findings on the scans. And what was reported is that just by having better imaging, there was a 25% absolute improvement in cancer control. No difference in the radiation, same radiation, but just targeted better. So this scan found new spots of disease that the conventional imaging missed. The radiation oncologist could then target those spots with extra radiation, or in some cases, they wouldn't have gotten any radiation to those spots, and that translated to better outcomes. So there's the old DuPont saying, better living through chemistry. Well, this is better living through imaging. And so another question, of course, is, well, if we can achieve a 25% improvement just with better imaging, I want to put that in context for you. There are very few new drugs out there that can achieve that kind of an improvement. Okay, so this is just an imaging test that can do what few drugs can do in terms of improving outcomes. So what if we were to take that same molecule, or a similar one, an even better one in this case, the PSMA molecule, and instead of attaching a weekly radioactive agent to it that emits weak radiation that can be picked up on imaging, and instead attach a very powerful radiation-emitting drug to it, and combine the two, image the patient, see if they express that molecule, and then give targeted therapy, with the idea being that this molecule is essentially the bloodhound that can sniff out the cancer. And then the radioactive agent that's attached to it is the Doberman that bites the crap out of the cancer, okay, and kills it. And so that concept was tested to great success on the Vision trial, which was published recently in the New England Journal of Medicine, and we were one of the lead sites. And the results were really spectacular in terms of improving outcomes. And, I could quote stats, but I think more important than that and more meaningful, because this study and the patients I treated on this study had a huge impact on me. So I want to tell you a story about a patient. And this particular patient, he came to me in a wheelchair. He was in his, we'll call it 70s, 80s, and had a cane that was fashioned out of a golf club. It was clear this man loved golf. And he told me, I have, two loves. He said, my grandchildren and golf. And then, he whispered, but golf comes first. And he was so unhappy in his life because he couldn't golf. And, He couldn't even go out to the putting green because he was in so much pain from his metastatic prostate cancer. And he said, "Doc, will this drug, get me to golf again?" And I honestly wasn't sure, so you don't want to overpromise. So I underpromised. And as it turned out, after a few treatments with this drug, not only was he up walking around, he no longer needed the golf club as a cane, but he was able to walk 18 holes of golf. And this was a man who looked terrible, and this drug completely turned his life around, and he's golfing and still doing well to this day. A guy who, seeing him, I thought he'd be dead in 3 months. So, that result really kind of got my attention and convinced me that, boy, we really need to focus more of our efforts on developing these types of treatments and doing these types of studies. And so at WashU, we're really fortunate because we have a lot of expertise, and this long predates my arrival, on the radiation oncology side, kind of measuring dose, delivering these types of agents and studying them, and then also on the radiology and nuclear med side, under the leadership of Dr. Rich Wall, Dr. Farod Dadashi, and of course you heard from Dr. Iravani. And so right now we have 4 clinical trials that we have open that we're running, looking at different agents similar to the one that my patient, the golfer, got. And we're really excited to try to develop new concepts to take these types of drugs and test them in patients with earlier stage disease. Once you're metastatic, we can treat you, but we really can't cure you. But these drugs, which are so effective in the metastatic setting, we might be able to enhance the cure rate for patients who have aggressive but still potentially curable disease. And so we're developing concepts where we're trying to find ways to give drug to enhance the expression of the molecules on the cancer cells, which are then targeted by these drugs. Looking at studies combining external beam radiation, hormone therapy, and this drug to try to boost the cure rate. And what I'm probably most excited about is that together with radiation oncology, and this is, with a lot of help, of course, from the folks I mentioned on the radiology side, but also in radiation oncology, my mentor, Jeff Mahalski, Dr. Mahalski, as well as Dr. Hallahan, to put together a collaborative theranostic center, co-led by both departments, where we can kind of leverage our strengths and our expertise in imaging and therapeutics and develop really sort of novel trials, novel research, novel ways of educating the future doctors of tomorrow, and to kind of put that together and be really the world leader in this field. And I think that WashU really has the potential to do that. And so certainly we would welcome, your thoughts and support, because I really think that this is an area where there's great potential and where WashU and really St. Louis can be a leader. So thank you very much for your time.
Okay. Can we have a few questions from the audience? Got to be a few questions. Yes, please.
Yes, Amir, the 25% improvement that you make reference to, is that due to equipment or is that due to the professional's knowledge of the situation?
Well, so I think it's actually just due to the fact that the patients who got that special PET scan, the doctors saw new lesions on the scan, which then the radiation doctors, so people like me, could then target. So let me give you an example. With conventional imaging, if someone has an early recurrence of prostate cancer after having their prostate removed, and their PSA is low, it's very unlikely that an MRI, a CAT scan, any of the conventional imaging is going to show anything. And so the standard treatment for those patients has actually been to just assume that the cancer is where the prostate used to be and radiate where the prostate used to be and basically cross our fingers. I mean, I wish I could say it any other, but that's basically what we're doing. And that, so this imaging study, say in this case, might show a lymph node that we otherwise would not have seen. And that lymph node has cancer in it. And so if we can see the lymph node, then we can treat the lymph node. And so instead of just aiming our radiation where the prostate used to be, we can treat where the prostate used to be and treat that lymph node to a high enough dose to eradicate all of the cancer where that lymph node was. And so if we can see things that we could never see before, then even conventional radiation can, achieve a better result. So, that's, I think, what's so exciting about it is the actual treatment didn't involve a new technology. It was just better imaging. And as sort of a student of military history, I mean, this makes sense to me, right? I mean, if the Roman consuls knew where Hannibal's army was, they wouldn't have been, massacred at Cannae, okay? If Rommel knew that the Allies were invading in Normandy on June 6th instead of Pas de Calais, he would have hurled our boys back at the beaches. But they didn't know. If you know, you can attack the cancer and completely change the outcome on the battlefield. And that's what we saw with this study.
I think kind of adding to that, what's important is the lymph nodes can be 1.5 centimeters, and Amir, you can chime in here, but one of the great things about these technologies are 1.5 centimeters, you can hide a lot of cancer cells in there. But with these new imaging modalities, you can actually start to see that, right?
And we can go beyond what actually the equipment is limiting us in terms of the resolution of the scanner, because it's highly dependent on the biology of the cancer and those cells, how densely they express those receptors. So we can see the lymph nodes the size of 2 and 3 millimeter, which are otherwise considered normal, but those are harboring cancer cells. So that's the progress has made treatments more accurate and more targeted.
Other questions? Got to have some good questions. We're going to lock the doors and not let anybody go out until you have some good questions.
Tim, there's a question from one of our online viewers. How long does it take for an idea to turn into a new form of therapy? Great.
Okay, the question, how long does it take for a new idea to become a new treatment? Anybody want to take a stab at that?
Stab at that. There's a concept that I had in my lab before WashU. I was at University of Kansas, and prior to that I was at Vanderbilt. So I had a concept of targeting these chemotherapy-resistant ovarian cancer cells with a combination of drugs, developed the concept in my lab at Vanderbilt. Fast forward 10 years later, we finally got a clinical trial. So that was actually relatively fast, and we're speeding up our clinical trials now, and we're doing parallel studies where it's going from bench to bedside and back. And so, I think that we used to think 20 years ago it would take decades to get something from the bench to patients, and what we're seeing is that we can do this almost in real time. And so that's the advance we've made in science. And so a concept could start, right now we're screening new drugs in our lab, and the hope is that we can take some of those drugs in the next 2 to 5 years into patients. But there are also drugs that are already existing, used for other purposes. We can immediately start a clinical trial if we have a proper target biologically. So advances are rapidly increasing our ability to take a concept from the bench to the bedside and back.
I think one of the great things about, I came from Stanford about 7.5 years ago, and they had a similar type of thing. More recently, WashU has developed the Scandalaris Center, the LEAP program, that really helps investigators take ideas and rapidly translate them and commercialize them. And of course, there's a Cortex Center, which is here, and it's just this great environment that really allows us to do that rapidly. Whereas if you live in the Bay Area or Boston or New York, there's benefits to being in St. Louis. Space, resources, and that you wouldn't normally have in New York. I mean, you can't have free lab space in New York City, right? So there are real benefits here, and I think WashU is such a powerhouse with respect to innovation, and I think what is happening is the translation of that into the commercial space. And so that was one of the things I really, has really been helpful, and I think is, I think will continue to grow as we see that.
Yeah, I often say, yes, ma'am.
How close are you all to figuring out who gets cancer and who doesn't? And what was done when they were 5 and 10 and 20 and 70 that we don't, and the people who do? Does that make sense?
So, yeah, great question. Great question. How do we know, or how can we figure out who gets cancer, what's unique about them, as opposed to people who don't get cancer?
And what the people who don't have done to not get it?
I think all the people who don't get it have a scotch every day. No, actually, that's a complex question, and I think there is no right answer. I mean, the obvious example is people who are heavy cigarette smokers. Their risk of lung cancer or esophageal cancer, other types of cancer, is remarkably increased.
Yeah, we had, again, my lab does tumor immunology, so I'm not a genomics geneticist. But working with Adil Chowdhury and Chris Marr, especially from Chris, we had actually started, I think we put together a small proposal, looked at this kind of phenomenon of genomic scarring. And so what we were looking at is in veterans who may have been exposed to Agent Orange and developed prostate cancer, for example. So there are methods to kind of look at the genes and see what kind of genetic insults, genomic insults have been accumulated over the years. And so certainly there are a number of people looking at that type of thing. And those aren't hereditary things. Those aren't things you pass from generation to generation. Those have been environmental insults to the genes that then lead to cancer development. But it's a fascinating question that's complex, for sure.
Yeah, it's a little bit of all those. Yes, ma'am.
So what would $100,000 or $250,000 do right now?
What would $100,000 or $250,000 do? It actually would, carefully invested, it could be the pilot work for actually each of these investigators. Because they all started out with a great idea, but you need proof of principle. And Russell mentioned the pay line at the NCI used to be 8%. So 92% of the grants are turned down. And the best way to have a grant funded is to have good data and proof of principle. So you'd take that $100,000 or the $250,000 and you'd invest it in one of the ideas of any one of these remarkable investigators, and they would show proof of principle and have preliminary data. And then they would take that preliminary data and proof of principle and submit it to the National Cancer Institute. And we get a $4 or $5 or $8 million grant. And I say in my lifetime, in the 40 years that I've been doing cancer treatments, we have come a remarkable way. Because at the beginning, when I was first trained, I was trained to be a lumper. If you had an ovarian cancer, we treated all the ovarian cancers the same way. And if you had a prostate cancer, you get treated the same way. And if you had a breast cancer, you'd have a mastectomy. And now we realize that all these things are many, many different diseases. There's 16 different kinds of lymphoma. And each of them are treated a little differently. They may receive chemotherapy, but they're treated differently. And that's happened in this 40-year window. And so what's really remarkable, I think, is you see these investigators and their teams that they've assembled at Siteman and Barnes and WashU, and it's made remarkable progress in a very, very short period of time.
May I give an example of
Please.
$250,000 of Frontiers Award to Dr. Catherine Fu, who now has NCI funding and R01 grant, which is the gold standard for being an investigator. She is now the lead, the translational lead on an international clinical trial based on the work that was done here through the generosity of Frontiers Award.
And I'll also acknowledge another, Will Galanders, who Russell mentioned. The concept of doing these personalized vaccines, Will did them in breast cancer, and again, came through a Cancer Frontier Fund grant. So, you know, there's a lot of these very carefully vetted investments. The Cancer Frontier Fund was really thought of as the concept was a venture capital fund. And so you very carefully invest these with the idea that you're going to get a much bigger return on your investment. And just the way Catherine did in her study with resistant ovarian cancer. So other questions? What? Ooh. Oh, well, we're going to alter the program. Dawn's trying to be the director here, you know, in the background, but, you know, but actually my boss actually wants the floor. He wants to make a few comments and then he'll introduce Jim, okay?
Thanks, Tim. Thanks to our guests this evening. I just make notes. People know I do this a lot. I sort of pick up on themes and I pick up on quotes, right? So here's what I heard. Common cancers that are underfunded, common theme. Need for funding to try new novel approaches. You know, the definition of the Cancer Frontier Fund for sure. New diagnostics for cancer treatment and cancer diagnosis. Novel vaccine approaches. Quite honestly, Siteman and WashU are leaders in this area internationally. Completely new immune players identified. Targeted approaches. A quote, one of only 10 in the country. A new lexicon, a new word in our lexicon, theragnostics. Who'd heard of that before? A mashup of typically diagnostics, radiation, and therapeutics that live separately, right, and more serially administered, now together, providing amazing results. And then I know Dr. Wall was smiling under his mask. Better living through imaging. I thought you were going to get up and cheer when you heard that. There he is. Dr. Rich Wall, the chairman of radiology. Yeah. So anyway, the other thing is I heard each one of our investigators, our clinician investigators, tell a story, a personal story about a cancer journey that they took with their patients. So the compassion that you heard from our investigators this evening is amazing. More importantly, their commitment to pushing back the frontiers of cancer and leading in these areas. So all of us have a role on that journey, right? And as ambassadors, you guys do amazing work getting our community behind this work and these amazing investigators at Siteman Cancer Center. Jim, would you like to bring us to a close? Thank you.
Thank you, John and Dr. Eberlein, and thanks to all of our physician scientists. who shared some of the groundbreaking work tonight. I can tell you it's incredibly humbling to stand up here after the display of talent that we've had here tonight as a layman and address you all. So I'm happy to be able to do that. What we just heard and the feeling of hope that it inspires is why I am so incredibly honored to be a Foundation board member and an Illumination ambassador. I'd also be remiss if I didn't tell you this is a little bit personal. I am a patient at Siteman Cancer Center and have received a series of treatments over the course of the last several years. Point being that the most recent one is something you can't get at any treatment center. Siteman is a qualified provider of what I need. And so for me to be able to wake up in my home in St. Louis, Missouri and drive 15 minutes to the hospital and get that kind of treatment and then leave the hospital that evening and go home and sleep in my bed that night is incredibly gratifying, and it's a blessing. And it's the result of everything that what you all do for the hospital. So I am very grateful for that. The work that's being done at Siteman is astounding, and the reason it's possible is because of philanthropy. It's that simple. Everyone who supports Illumination is a part of the progress that we heard about here tonight. Speaking of support and philanthropy, I would like to make, and John referred to a special guest that we have here. I just want to point out that Ruth Siteman's in the room tonight, and I think we should give her an extraordinary round of applause. Thank you. I hope you'll all consider joining us at the Illumination Gala on June the 4th. It's at the Ritz-Carlton. For the first time in 3 years, we're going to be in person. It's going to be fantastic, and I think it's going to be very well attended. We're shooting for a big number. If you can't join us, please consider still supporting the incredible work that's happening at Siteman Cancer Center. Please ask a Foundation team member who would be here tonight, or visit the Foundation website for more details, to look for the details of the event, and also for your invitation, which will hopefully be coming in the mail soon. That concludes our evening. I would invite you to all stick around and have a drink and something to eat afterwards and visit with our esteemed guests here tonight. And I thank you all for coming, and have a wonderful night. Thank you.